Going over oxidative phosphorylation and the electron transport chain. To start with, we will quickly describe a mitochondria. This in the red is the outer mitochondrial membrane. It consists of a phospholipid bilayer. And in the blue is the inner mitochondrial membrane. It also consists of a phospholipid bilayer, and this is where oxidative phosphorylation takes place. In between these two membranes is the inner membrane space, where you'll see hydrogen ions accumulate throughout this process. On the other side of the inner membrane would be the mitochondrial matrix. This is where the Krebs cycle occurs, and it produces NADH and FADH2. So, I have decided to use cookies since this all starts with glucose. There's plenty of glucose in cookies. I'm just going to demonstrate one NADH and one FADH2. However, in the actual cycle, there is 10 NADH that will go through and 2 FADH2. 2 NADH are produced during glycolysis. 2 are produced when pyruvate is converted to acetyl coenzyme A, and 6 are produced during Krebs cycle, as well as 2 FADH in Krebs cycle. The electron transport chain is a sequence of proteins called chitosomes. Chitosome B, chitosome C, chitosome A, chitosome A3. Each chitosome contains a protein and a pigment. The pigment contains a metal, which is either copper or iron. NADH is a reduction of NAD. Here you see NAD, here you see NADH. At this point, either NADH or FADH2 will give hydrogen ions to a coenzyme. Now this is where they're different. If it were to be NADH, it'll give its hydrogen ions to coenzyme flavin mononucleotide, abbreviated FMN, or if it was FADH, it'll give its ions to hydrogen ions to coenzyme Q that can also be called ubiquinone. So, for this purpose, we're going to use NADH. When NADH gives its hydrogen ions to FMN, it becomes NAD, which is an oxidation of NADH. An oxidation is when it loses ions. So from here, FMN will go to cytochrome B, or protein complex 1. The electrons slowly lose energy as they pass through the cytochromes. So now, when this happened and it released the 2 hydrogen, the hydrogen go into inner membrane space. From here, the electrons go from cytochrome B to cytochrome C, where 2 more hydrogen ions are moved from the mitochondrial matrix to the inner membrane space. From cytochrome C, it goes to cytochrome A, and from cytochrome A to cytochrome A3, where in that process 2 more are moved from the mitochondrial matrix to the inner membrane space. Now, the 2 electrons are tired out from their long journey and are low energy, where they are accepted by our oxygen that is waiting right down here. So here's our oxygen, here's our 2 electrons, and now 2 more hydrogen come along and they combine with this oxygen, and now we have water. So these can move out the way, and here's our water. From there, technically the electron transport chain does not produce any ATP, but instead it creates a concentration gradient. As you can see now, the most hydrogen and protons are in the inner membrane space and not the mitochondrial matrix. Therefore, this gives their extra energy for the ATP synthase, which is another protein, to go from ADP and make ATP. This process is called chemiosmosis, and the ADP is adenosine diphosphate, which then gains another phosphate and becomes ATP, adenosine triphosphate. So if it was NADH that went through this whole process, 3 ATP are produced for every 1 NADH. If it was FADH2 that went through this process, it'd be 2 ATP produced. FADH. There is a weaker concentration gradient, therefore less energy to change ADP to ATP. So from the electron transport chain, 34 ATP can be produced from the 10 NADH and the 2 FADH.